Okay, so let's start. I think I got too excited with too many questions of this quiz. Yeah, I'm going to see if I curve it or I remove it because when I thought that I had like generally have 5 or 10, there are too many questions. So if you don't do well, I will remove it. If yes, I don't want, the idea is not that you get a bad grade. The idea is that you show me that you were in class. Okay, so just to let you know, if you want to see like if you some of you will be very happy with the grades of your test. Yeah, most of you did very well. Other ones, if you have a question, you can come and or ask for an appointment, and we can see. Maybe I had a, after counting so many times, maybe I went 21, 22, 11. I made a mistake, and I will be more than happy that you check. I try to do it, but there is a point that I just see numbers and I forget things. So we can do it after class, or we can do it when you have time. Okay, you can just email me and we can check that. Tomorrow, we are going to meet again downstairs for the lab, and we are going to, please, the idea of the lab tomorrow is that you work um, with the reference manager. So instead of you typing all your reference, now there's a software that do it for you. If you want, you can watch all the video that I posted there, and you can do it even from home and just do it come tomorrow very quickly. Show me that you're able to do those things, and if you watch the entire video, I put like how to install the software or how to and how to put it in Word. If you want to watch, it's like 20 minutes, and you will learn all the things that you need to do of how to drag the file into the scientific file and scientific article into the software, and then how to tell Word, turn this into a reference that is just click here, and click here. Hopefully, it's so if you want to watch it and you want to do it at home and you want to show me, or you want to show me the question that you have, feel free to do it tomorrow, and so you can do it much faster. All the steps are there, and you can give a look. What else I have to tell you? Please, I know that one of you will have a terrible time with the software tomorrow or tonight. It always happens that there is a computer that have a glitch. Why? Because computers always have like that. Relax, we will try to solve it tomorrow. If you are the unlucky person, sometimes it is very easy. If not, you can work with somebody else. There is, in theory, all those software are they work for all the computers. However, there is always a glitch that appear. So don't get stressed. And that's all. So let's start. We're going to talk about 4 things today. The first thing that we're going to talk is the endomembrane system. Yeah, so we're going to give a quick review about the endomembrane system, and we're going to focus on this, the Golgi apparatus. And one of the main function of the Golgi apparatus that is there in the ER is the process of glycosylation. Okay, so I'm going to tell you how proteins you add carbohydrate, that this is the most common post-transcriptional modification. And then I will show you that another thing that the Golgi apparatus does is add tags. Yeah, so it's going to produce all these vesicles that go to different parts of the cell, how they're able to the lysosome, how they are able to the membrane, because they have a specific tags. And at the end, I will tell you how some of these vesicles, they go outside of the membrane. Yeah, and for example, the insulin is not going to stay in the cell, it's going to go outside. So we are going to talk about that, about these secretory pathways. Okay, so 4 key ideas. These are the key topics that when you need to study for the test. And we are going to start with the endomembrane system. In the endomembrane system, there are only 3 key ideas that I want that you remember. First thing, do you see this Golgi complex? In the same way that this room has a up and down, yeah, you have left and right. Yeah, there is a polarity. The Golgi apparatus has the Golgi complex has a polarity that has a cis phase and a trans phase. And this is very important for the function. Cis phase will be the molecules that are immature go in the cis phase, and the ones that are mature will leave to the trans phase. Okay, that will be fundamental for you to understand. I'm also going to talk about the models. We still don't know how the Golgi apparatus work. You need to remember that there are two models. One in which the vesicles move and they move actively. Yeah, like this person that is moving to the stairs. Yeah, this is an active movement. Another model, yeah, we are not sure which one is true. Maybe both are true. The vesicles are moving passively, like when you are in an escalator, you are not moving, the escalator is moving. So what we think here is that the Golgi apparatus is moving and the vesicles are not moving. So remember, Golgi apparatus has a polarity, and the Golgi apparatus has two models to mature the vesicles. And finally, if all the vesicles are moving from the ER, then to the Golgi apparatus, then to the membrane, you are going to remove phospholipids all the time, so you end up with a giant membrane, and the ER will disappear. So yeah, and all the enzymes will go there. So you need to recycle those. Yeah, so the vesicles, they have a movement that go forward, but they also have a movement that go backwards. Yeah, that is the retrograde transport. So good. So three things that I'm going to tell you about this. So let's start. Okay, so Eric, sorry. Each of this you have heard about all this question multiple times, slightly different variation. The endomembrane system has different components, except the Golgi complex, no. The endosomes,
That's part of it.
yeah. Lysosomes,
Yes.
perfect. Peroxisomes,
Autism.
no. Good. So in the test, that is correct. Have you heard about this one?
No.
Yeah. So in anatomy and physiology, you hear that sometimes the epithelial have variations and they have glands, yeah, and some glands have like pipes and they remove, you through those pipes, those cells that you can expel, some uh, different products, yeah. So one that is very famous is this one, this cyanogen granules, because this is where the insulin come from. Yeah. So this is part of the endomembrane system because it's a specific type of vesicle that we were taught in the class. Okay. So this sounds weird. Yeah, but the correct answer. So the exception will be peroxisome, and this is also part of the endomembrane system. It's just a weird type of vesicle that is very important. That I think the last slide, when you are all tired and I don't remember my name, I will show you a slide. Okay, so just in case that appear in the test, you know what is that one. Okay, so the answer perfect, peroxisomes, and we had last class we talked about the ER. And you are tired that I tell you there are two ERs, yeah? And I just gave you the slides there in case that you want to study. And there are three functions that in your notes you wrote it. The functions like detoxification, the metabolism of carbohydrate to go from a polymer to a monomer, and the calcium storage, okay? And you can read more of that in the textbook. If you have any doubt about that, we also have the rough ear, and I told you that they are in charge of organize the proteins, that they form the disulfide bonds, that they are the one that make the protein more stable, and also they are in charge of the quality control through the chaperone proteins. And today we will talk about the glycosylation, that it start in the ear and then it continue in the Golgi apparatus. Okay, so this is the time that you that we just need to know the different components of the Golgi complex, yeah. And here, this is the typical slide that the typical animation that you will see about the Golgi complex that look like pancakes, yeah, stacks like that. And here there are 6 components. And so you see very clearly here vesicles. Let me see if I can. Here you see the vesicles. Here you are secreting some vesicles. Yeah, they are just, this one is a new vesicle that is being secreted. Yeah, it's going to detach from there. And the lumen will be this green inner space. I will rotate it a little bit to produce that, to see it clearly. And again, this external, it will be the external component like in the ER. And now you have this trans and cis phase, okay? That I will show you in the next slide. So the cis phase is the one that is close to the ER, and the trans phase is the opposite to the ER. So let me show you this here, yeah? So cis phase is the one that is close to the ER, where this molecule will go immature there, and the trans phase will be here. That will be away from this one. So just from the roots, cisgender, you know, biological sex and gender are close to each other. Transgender, you know, the biological sex and the biological gender, they are far. Yeah, they are not. So here the root is the same. Yeah, one is close to this one, the other one is far. Okay, so here cis will come the immature vesicles. Here the mature vesicles will go to the trans. Okay, and we will be talking about that during the class. Okay, so here again, I just rotate this animation in different ways, just here to show you the lumen that is the internal space of the cisternae. Okay, so here in this video, they just summarize the function of the Golgi apparatus. And you cannot hear, yes? Let me see if it's only. Oh, anyway. So the Golgi apparatus has these functions that are the production of glycoproteins and sorting the proteins and the lipids. The anatomy is these flattened membrane bounds, and they have a disc shape. Okay, this you have heard about all these things before. Just wanted to show you a video from the textbook, but right now I don't want to mess up with all the settings of the computer. Sorry. So Rico, could you read this question? Remember, there is polarity. Cis is the one that is close to the air. Immature molecules come there. Trans is the opposite. So where the mature molecules go there. So which one do you think that is the correct answer there? Duncan, any help? What do you think? So here, Asher, which one do you think that is the function of the cis? C. See why? Because it's receiving is close to the ER, so it's receiving all the proteins and lipids from the endoplasmic reticulum. Okay, so here, if this appear in your quiz, pretty easy. Remember, is this one? Sorry, is C. Okay, the cis Golgi is the one that received the proteins from the from the endoplasmic reticulum, the trans Golgi, it will be the one that transported to the cell membrane. So here you have the Golgi apparatus, or the Golgi complex, is formed by cisternae, and there are three components. Yeah, if you check your textbook, so the cis Golgi is the one that is close to the ER. The mature molecules go there. So here you see cis Golgi network. They come here from the ER. So they arrive from the ER. The next one is the medial cisternal. Yeah, here they will be processing products. Yeah, you have a lot of enzymes that are going to remove or add lipids and also carbohydrate there. And so here's this. So cis, medial, and finally the trans Golgi that it will be here. Okay, so you have these three components of the Golgi apparatus. Again, the Golgi apparatus or Golgi complex has a polarity that is important to understand the function. So perfect. So here the trans Golgi is in charge of sorting and packaging the proteins. The modification and processing of proteins occur in the medial cistern, and the cis is the one that receives the proteins. This one transporting to the cell membrane. You know, actually, this one now that I think here what they are referring is that the vesicles are the one that are moving the proteins outside of the Golgi apparatus and send it to the membrane. Yeah. So this one, the actually a trans Golgi is the one that is sorting and package those one to later send it here. Okay, now it's clear. So three components, each one has a different function. Yeah, and this one, the last one, is the one that send the vesicles. Yeah, here is the vesicles that they go to the membrane. Okay, so here you have two models. We don't know how the Golgi apparatus actually work. This will change in your lifetime. We think so. We have confocal microscopy that we can put GFP and see how the vesicles move, but we don't know if maybe the two models work at the same times, or we don't know if, for example, in plants one work and in animals the other work. So here you're moving the lipids and the proteins, yeah, and through the Golgi, and we don't know if there is one is called the stationary cisternal model, and the other one is called the cisternal maturation model. Okay, so two different models. And it sounds complicated, but actually they are pretty easy. Again, oh, don't tell me. Okay, let's try again. Sorry. Now I understand what was the problem. The cable was one millimeter detached, and that was all the problem. I thought that, okay, let's see if Okay, so two models. What they explain in the book, in the video, and you can also read it in the textbook, is that we don't know exactly which one is, but there is two models. In one, there is a stationary model. So here the cisterna doesn't move. To me, it remind me when somebody is going upstairs, the vesicle is moving, the person is moving, the stairs are not moving. So the Golgi apparatus is not moving. The other model that they show you, yeah, is this one, that this external maturation model. In this case, the Golgi complex is the one that is moving. So there is a passive movement of the vesicle. So the person is not moving, is the Golgi apparatus that is moving. Okay, so two type of movements. Yeah, we don't know which one is working. We just know that they could be happening any of those, and we will solve it in future years. And so what they were showing you in the video is that you have vesicles that are moving through the cistern. In the vesicles, you have proteins, yeah, but in addition to that, you have enzymes. And those enzymes, imagine if you are moving all the enzymes from one to the other one to the next one, yeah, this will help the proteins to be folded. They will help it to have to get glycosylated, that I will show you that in a second. But they also, if they end up living in the vesicle, if they will be outside of the membrane. So those one will need to be recycled. And I will show you that in a second. So what I show you is that, and in the video, that you have shuttle vesicles, yeah, that they will go from the cis here, CGN, this cis, this C is from cis Golgi network to the T that is mean trans Golgi network, yeah, and they will start moving. This is a disassemble, go back, and all these enzymes will move with the proteins. And something very important is that you will have tags, yeah, that are able to retain those enzymes, yeah, or retrieve those enzymes. So what I was telling you is that imagine that all the enzymes end up leaving the membrane. That will not be useful. So they have a specific tags, so the enzymes go back. Okay, and we will talk about that in a sec. But this is the other model. That is the maturation model. That the thing that it moves is the Golgi apparatus. And each, as you saw in the video, that they change the color of the Golgi apparatus. Yeah, they put it red and they move it here red. Yeah, so here what is changing is the Golgi apparatus, and the enzymes that are there will start also changing. One get activated and the other one get inactivated. So enzymes no longer needed in the late compartments. They need to go back, yeah, to early compartments. So again, two models. Remember, they will need to have enzymes. They need to move phospholipids, and we don't know exactly what they which one of those could be both. So I just told you as a summary that these two models might not be mutually exclusive. Right now, we just know that maybe both could apply, and they could apply to different type of in different type of organisms. And every article or video that I watched, they talk about this. They said that this could change in 10, 20 years, that we can decide which model is correct in which organism. Okay, so I told you that there was a polarity, cis and trans, and now in the test, you know which one is the function. Now you know there are two models. One that the Golgi apparatus is not moving, the vesicles are moving, and in the other one is the Golgi apparatus that is moving. And I show you one person going up the stairs, and in the other one, the escalator. Good. And now I'm going to, I told you that the recycling is very important. Why? Because you're moving lipids, you are moving enzymes, you're moving proteins. And if you remove all of this, the ER and the Golgi apparatus will disappear and the membrane will get giant. So these that go through exocytosis, yeah, you have mechanisms to recover that. So that recycling, that movement, so this anterograde movement is the front movement that you can see here. Yeah, that go in that direction. That is go from the ER to the Golgi apparatus and then go to the plasma membrane. You need to balance that with the opposite process. Yeah, so this will lead to the exocytosis. And the other movement, that is the back step that you can see here in purple, is called retrograde, and it allowed to balance the flow of lipids. It allowed that ensure the supply of components, so and it will recycle lipids and proteins in the late stage of the anterograde transport. So all of this is not lost; it went back. It goes back into the early stages. Okay, so I just told you 3 things that I wanted to remember. The Golgi complex have 2 phases, cis and trans, and those are very important. I also told you that there were 2 models, one in which the Golgi apparatus don't move, the vesicles move, and I told you that the other model, the Golgi apparatus is the one that move and the vesicles don't move, and that the vesicles, they need to be recycled. So you have one forward movement, go in this direction, but you also need to recycle those enzymes and phospholipids, so you have a backward movement or retrograde. Okay, so this is the first. So I just told you about the endomembrane system. Now we're going to talk about the function of this endomembrane system that happen in the ER and also in the Golgi apparatus, that is the protein glycosylation that we have been talking before. In case that you need to study, these are the topics. And I'm going to tell you what is the protein glycosylation that all of you already know. And I will remind you, okay, sorry for the typo, this is the most common cross-transcriptional modification. I will show you the different steps, how you put the carbohydrates into the protein. So if you remember the process of translocation, yeah, you put the ribosome inside of the ER, you have to do the same process of translocation, but with the sugars, with the carbohydrates, to attach them there. And there are, I think, 5 or 6 steps. There are different types of process of glycosylation. And yeah, here we have 2, the end link and the O link, okay, that we have talked before. So I will remind you that. And finally, at the end, I will show you that this happens, some of this process happen in the ER, but there are a lot of processes that also happen in the Golgi network. So this glycosylation will start in the reticulum endoplasmatic, yeah, but it will continue through the ER, okay? In all these vesicles that I show you, they are moving. That is what is happening. Some enzymes are doing those glycosylation. So again, glycosylation is the most common form of post-transcriptional modification. I think many lectures ago, Maddie told us, they are very important for the cell recognition. And definitely this is a very important function. So here you have the membrane that we already covered. You have proteins and you have the glycoproteins. So the blood groups that we have, all of us, they are an example of a protein that have a specific type of carbohydrates, okay? And they have a covalent attachment, yeah, so the protein to the glycan. But how this happened? It happened in the ER and happened in the Golgi apparatus. And if you go to your textbook, you can see that there are all these steps, yeah? They are not complicated. You just have to take some time to read it, okay? So I'm going to show you how they are able not only to form these carbohydrates, but to move it from the cytosol and to put it inside of the ER and attach it to the proteins, okay? Oh, sorry. So the steps are you're going to have a carrier protein, a carrier molecule, sorry, that it will help you to move the sugar. So you're going to put the sugar there. You're going to flip the sugar inside of the membrane, yeah, in the carrier. Then you move it to the transfer to the sugar, and then you're going to remove some sugars. Yeah, so I show you in a schematic. Now I'm going to show you where your textbook does, but they're generally these steps. Yeah, carrier, adding sugar, flipping it inside of the membrane, and transferring. So first step, our carrier protein, our carrier molecule is this dolicol, yeah, that here it has a phosphate. So here the phosphate group is here. It will work as a hook. Yeah, it will attach here, the carbohydrate will go here. This is the part of this alcohol, the dolicol here. And this is in the textbook, they instead of showing all these long molecules, they just write that. Here is the ER, and here in orange will be the cytoplasm. Okay. So now, second step, you have the dolicol molecule. Here is the cytosol. So what you need to do is to form the sugar. Yeah. And here I show you 3 steps that they show in your textbook. It starts in the cytosol, in the cytoplasm. Yeah. And you are going to add different type of sugars. Here in blue, you see this N-acetylglucosamine that is there. And you are also going to add this in green, the mannose. Yeah, so you have the dolicol, and you start forming this by adding different type of sugars. Okay, so that you see in different colors. Okay, and they are added to the dolicol. So now, last time I told you there was a process of translocation. So you take the protein inside of the ER. We're going to do the same thing, but with the sugar. Here, Angus, you are not going to remember the name of the protein. Do you remember the protein that it was involved in the translocation? That it has a number very similar to translocation? Yes, I will not remember. If you ask me in December at the end of the class, I will be like, I don't remember anymore. It was called translocon. Why I'm asking you that? Because that is only for the ribosomes to put the protein here. For the sugar, there is this one. Could you read it for us? I don't.
Let's,
Yes, yes. So it's going to flip. You see that the sugar is here, and now you flip it here. You know, in the computer, and now I know why some of you just look at the computer. It looks so pretty. Here the focus is not very good, but yeah, excellent. The flip paste is going to flip the sugar and is put it inside. So it's going to translocate it. It's going to take it from the cytosol and is going to put it inside of the ER. Okay, so it's again a process of translocation, moving from the cytosol into the ER, but we are using a different proteins. Okay, so here, again, last time I told you that the protein translocation is the process by which proteins are moved between cellular components. In this case, we're doing the same thing, but we are moving is the carbohydrate, and we are moving it inside of the ER. So it goes from the cytosol to the ER lumen, but we use as a different protein that is the flipase as Angus told us. Yeah, and still the dolicol is holding this carbohydrate there that is forming. Okay, more steps. Here is pretty easy. If you see, we have all this phosphate, blue, different type of sugars, yeah, in blue and in green. And now we are going to just add more sugars and add other type of sugar, not only in green, but also in purple, that I guess that one is glucose. So this one occur already in the lumen of the ER. You added more mannose sugar that are here. You see that there are many more green components. Yeah, you added more sugars, and also you add some glucose. Okay, and here you see the sugar. Now what you're going to do is to remove. You remember that I put a carrier protein? Yeah, something that is carrying. Now you're just going to move it from the dolichol, and you're going to put it in the protein. That is the last process of the glycosylation. So the dolichol released this, and it moved into the aspergine in a protein. Okay, so here you transfer the complete oligosaccharide, yeah, that you have it here, and now is not going to be more in the dolichol here in purple. Is going to go into a protein that you see here, and this is catalyzed for using this protein oligosaccharide transferase. Okay, and of course there are more steps. That those steps include removal of some sugars and adding new ones. That those processes happen that happen in the ER before this is transferred to the Golgi apparatus. Okay, sorry. So here you see you have the mannose here and the glucose here. You're going to remove this one here. Yeah. So again, it's not complicated. It's just a little bit of steps. And this is what happened during glycosylation. So remember, you have the carbohydrate outside in the cytosol, you form it, you flip it, and then you add more, and then you transfer it to the protein. And now that they are attached to the protein, you can remove some of those sugars. Okay. So I already told you what is glycosylation. Then I told you the steps of the glycosylation. Now I'm going to tell you two types of glycosylations that you already know. One, that is the end-link glycosylation, that here you have just, that is attached, the polysaccharide is attached to the nitrogen here. This is what is end-link, and is found in eukaryotes, and is found also widely in archaea, yeah, and is rarely found in bacteria. Yeah, you can and here is attached to the nitrogen atom. As I show you here, let me in the nitrogen here. This is why it's called end-link. The other one is the O-link, that you are adding to the oxygen that is located here, and you can find it in various processes like cell signaling, determine the blood types, and also giving stability to the proteins and ensuring the proper folding. Okay, so there are two types of links, one that is the end-link and the other one that is the O-link. In addition to the glycosylation of the ER, yeah, that happened, that I just showed you here, you have also glycosylation in this process in this cis Golgi apparatus, and then it continue in the medial cisterna, and so the glycosylation starts in the ER, but it continues in the Golgi apparatus, and it goes all this way. You will have specific enzymes here for the ER, and you will have also the specific enzymes in the ER that are going to produce all these functions. I don't think that I will tell you more about all the enzymes. I just want that you understand what is the glycosylation and the different process. Okay, so this is the half of the class of this class. So you already know what is the process of glycosylation. You saw the steps. You know there are two types, and you know that these occur in the ER, and there are more processes happening in the Golgi apparatus. Now I'm going to tell you about the how the proteins traffic the ER. So first thing that I will tell you is that there are hundreds of vesicles, so you need to have a specific tags. Yeah, and those tags will tell the proteins, the vesicles, where to go. Yeah, and I will show you that there are two types of tags. One that are the retrieval tags. So similar to a frisbee, you are going to send it and they will come back, and the retention tags are the ones that they tell the vesicle you are able to leave when all the process are completed. Yeah, so the retention tags will not allow to the vesicle to leave. Okay, so two type of tags. And then I will give you an example that is the of a process where you use this tags, that is the mannose 6-phosphate. You can read in more detail in the textbook, but what I wanted to tell you is that it reminded me of this of a needle in a haystack. You have hundreds and hundreds of vesicles. If you see here, hundreds and hundreds of vesicles that are moving. So how, like, for example, if I take sometimes I'm too tired. For the people that took anatomy and physiology, we finished so late, like at 8 or 9, and it was evening and it was winter and it was minus very cold. Yeah. And sometimes I say, like, I'm going to take an Uber, and I was here, but the Uber ended up going to the other place, and I was with my phone, and I was in the middle of the snow looking for the Uber. Anyway, how we with a cell phone are not able to locate an Uber, but molecules and vesicles are able to move to the right organelle is incredible. And one of them is to use these sugars, these tags that I told you. So the process of glycosylation, very important. And I'm going to show you how they go from the ER to the Golgi to the vesicles. And we are almost done. I know that I'm trying to summarize this with you. This is, and I'm sure that this in the future, this will be an entire there will be so many more examples later. But anyway, I just told you this. We don't know how vesicles move, and it's really complicated. And I can tell you the experience with the Uber. You can also tell me the experience. I don't know if you have your bags send it to a completely different country in an airport. Yeah. So but we have tags that help us to identify that, and the cells are very good to do that. So how the vesicles are tagged? You put on the vesicles short amino acid sequence, you put some sugars similar to the one that I showed you before, or you can put in the hydrophobic regions of the proteins or lipids. You change in those sections to form a tag. And one example of this again is this mannose 6-phosphate. And this will be in the test. So the slides are there. I know that this time is you're already tired. I'm going to quickly go through that. So I put this one because this is a retrieval tag. This is what is happening. So you're sending some from the ER an enzyme. It needs to come back. So you need to have a tag there. Okay. So you have to retrieve those tags, and you put tags here in form. So there are some that are called retention tags and retrieval tags. So here in a vesicle, you have not only lipids, but you can have different proteins, different enzymes that allow this vesicle to go from the ER, for example, to the mitochondria. Okay, and in a second I will talk about this retention and retrieval. Again, I put this so you remember two type of tags. This is the ER, and you already know that. Sorry to tell you this, there is a cis phase and a trans phase. So the molecules are coming from here, yeah, immature, yeah, they will come from the ER, and they will fuse to the membrane, and based on the membrane, they are sorted based on the destination, and the molecules undergo remodeling. And I show you one example that is the process of glycosylation. And then they can go to an organelle, or they can be secreted outside. Okay, so a question. How do you see that the cis phase is smaller than the trans phase? Yeah, tiny membrane, large membrane. How, Derek, how do you think that you can make a membrane small and another membrane much larger. So you have phospholipids in the membrane, yeah? So do you see this tiny phospholipid with the side chain that is very small and this one that is very long? So one way to store all these molecules is to put here just fatty acids with short phospholipids, and here you can have one with long phospholipids. You can also have more different type of lipid, more cholesterol, and this is why always this one is smaller than this one, that it will be larger. Okay, and this allowed the Golgi apparatus to modify those ones. So this hydrophobic region length, so this one will determine the membrane layer of the Golgi apparatus, and the membrane thickness increases from this cis Golgi network to the trans Golgi network. One of this is just that you have longer molecules and you add more lipids, more complex lipids, and different proteins that are larger. And these allow the sorting of these proteins and lipids. Two type of tags. Here, I just wanted to show you that one type of tags here, this person is not able to move. So the same thing happened with the vesicles. The immature vesicles, they don't move. And when they are mature, they are able to move. That is important. So only when these retention tags, only when they are completed, are able to move. And also, so those tags are very important, and we will show you one of those that it will definitely appear in the test is the KDEL tag, and I will show you that in a second. That is a retention tag. The other one is the retrieval tags. Adriana, this was a question that I had. What is a soluble protein? What is a soluble protein? And I was like, when I was going to teach this class, I wrote that, but I'm like, what is a soluble protein? What do you think is a soluble protein? So we have been talking in the previous chapter that you have proteins that are located in the membrane, yeah? The soluble proteins are the ones that are inside of the vesicle of the cytoplasm, yeah, that they are not attached there. So you can have tags in some of those proteins that are inside of the vesicles, okay? And I will show you also another example of that. So remember, one of these will be here. Write this down, COP1 and COP2. Yeah, write it down. This will be in the test, and it will be in the next lecture too. Here you have that when a retrieval tag will happen when a molecule goes from the ER onto the Golgi, and if it doesn't need to, that is not supposed to leave the ER, so you have a retrieval tag, so it goes back through the COP1, okay? So very important, COP1 and COP2. Two type of tags, retrieval tag and retention tags. And here I'm just going to show you, and I'll finish with this. This mannose 6-phosphate. So again, imagine hundreds of vesicles living here. So what we're going to do, we're going to have an enzyme, these lysosomal enzymes that are only going to go to the lysosome. And you have thousands and thousands of vesicles. So you're going to add a specific tag that is going to lead that all these vesicles, yeah, all that have this tag, yeah, that have those enzymes are going to go only to the lysosomes. Again, this is like a needle in a haystack that it's impossible to know how all these vesicles are moving to a specific place. One way to do that is using these tags that I show you. We are just putting a sugar there. If you remember the hydrolases that are located in the lysosomes, are just using, they have if you have a molecule and you add water and you add these hydrolases, it allows you to break this molecule, and you can put, for example, break it into the the molecule that one component with the hydrogen and the other one with the hydroxyl. Okay, and those enzymes are very important for the lysosome. Okay, so remember, what is this mannose 6-phosphate? It's a unique tag marker that allow the lysosome enzymes to go only to the lysosomes. And this type are on an end-link oligosaccharides that are located in these hydrolases. So I'm going to show you that it goes from the ER, then go to the Golgi, then go to the vesicles. Okay, so first thing, it started in the ER. Yeah, here is in the ER. You have the rough ER in the step 1. You have the enzymes that is synthesized there, and then you add a carbohydrate. So you have the enzyme, and you start the glycosylation. So the process of glycosylation that I show you happened here too. Then we are going to move into the cis-Golgi, that is the network, when the vesicles, these proteins, this vesicle are immature. So in the cis-Golgi is the step number 2, and you're going to add to the carbohydrate, the mannose. You're going to phosphorylate it using 2 different enzymes that we are not going to talk right now. So here you have, in addition to the carbohydrate, you have the enzyme, and you also have these phosphorylated mannose. Next step, you are not anymore in the cis-Golgi, you're moving to the mature Golgi, the trans-Golgi network. And here you are going to have a receptor. Okay, so here you are going to, this receptor are packed and transported here. So here you have the enzyme, the mannose, the phosphate, and it's going to attach to this receptor, and it's going to be ready to leave the molecule. Okay. Step number 4, they're in the early endosomes, and here you're going to have a change in the pH, and it's going to cause a dissociation from the enzyme to the receptor. So here you see there is not anymore the phosphate group, there is no anymore the receptor, you only have the sugar, the mannose, and the enzyme, and look, the receptor is here. Final step, there is a recycling of the receptor goes back, and is recycled, and the enzymes go to the lysosome. Okay, so it's located in the late endosome, and you have a receptor that is recycled. So I'll just show you that how some molecules, some enzymes can go to a specific place from the Golgi apparatus. So here they can go or to the late endosome or to the early endosomes, because they have a specific tag, that is the 6-mannose tag. This is very important. In the next lecture, we will talk about the process of endocytosis. So here the early endosome is going to go and form a late endosomes, and here you're going to have all these enzymes that are going to reduce the pH, and they are going to be able to degrade a lot of DNA, RNA, different organelles, all these macromolecules. Okay, so here you're just merging the vesicle to the Golgi apparatus here, and here you're not adding, you add the lysosomes to the lysosome, these enzymes. To finish, this will be in the test 2. Remember the importance of the pH. So I just show you that from a small change from 6.4 to 5.4, So from here, between the still water to black coffee, sorry. Yeah, is that is enough to produce a chemical reaction? Yeah, that here you have a receptor that is attached to the phosphate, and here you dissociate it. Yeah, so you will see that many times just changes in the pH are able to regulate how these enzymes lose, attach or deattach one of these receptors. Felicia, so could you tell me the changes in the pH from the ER to the Golgi apparatus and the vesicles? You can, I know what you're seeing in your computer is much clearer there, maybe. Could you tell me if there is a change or not? Yeah, so it start like neutral, yeah, around 7 in the Golgi, in the ER, and in the Golgi, it start getting is going to 6 to 5, and then in the vesicles it start getting lower. Yeah, so this is a mechanism that allows the dissociation to the receptor, and in the endocytosis, if you see something similar happen, and the lysosome is the one that have one of the most the lower pH. Okay, so sorry, for the dry class, there is so much things in the following lecture that we will see. But I just finished telling you that one of the main functions of the Golgi apparatus, yeah, is to tag some vesicles, yeah, to allow them to go from one place to the other one. So I gave you an example of the Uber that are difficult to move, or the bags to go to another place. Yeah, to go to a specific place, they use amino acids, sugars, and also they add in the lipids, in the hydrophobic regions of the membranes, or the proteins, you can add things there. And you have two type of tags, one that are the retention tags and the retrieval tags. And I show you one example that summarizes everything that is for sure going to be in the exam. That is the mannose 6 phosphate. That is, you have is only for lysosome enzymes and go from the ER, the Golgi, to the vesicles. Thank you for being awake. We made it. In the next class, we will see this. I have been telling you that we go from the nucleus to the ER to the Golgi. I already told you that there is COP1 and COP2. We're going to talk more about that. These are retrieval tags. Remember, here, if there is something that go to the Golgi complex, and no, you have to go back. I told you about this, the lysosomes, how they form, how they go to the mannose, and we will talk about this, the secretory pathways. Not all the proteins are going to go to organelles. There is a lot of them that go to outside of the cell, like the the granules that you have the insulin that we start the class. We're going to start talking about those ones. Okay, just take the quiz just as a practice. If you find a question that I didn't talk about it, I will check the quiz and or remove it. If you have a very low grade, if all of you got low grades, and if you got a high grade, I will keep it. Okay, so sorry, I got too excited with too many questions. Next class, we will talk about the movement of the membrane. And if you want to see the test, stay here and we can give a look, or we can give a look later.